Thank you very much and welcome back from lunch. Hopefully you didn't have too many carbohydrates because I need to keep you awake for this session and also for the next one because I'll be taking the next one as well. First of all, I am passionate about inhalation product. Hopefully you have got some interest as well. And if anything, you'll find some good tips for review inhalation products. As Michael said, I work in the quality office, so I will be covering the quality aspects of this session. And if you have any questions also afterwards, let me know. I will briefly give the floor to Sarina, who is online, so she also can introduce briefly herself, and then we'll get started.
Thank you, Claudia. Michael introduced me already, but thank you for giving me the opportunity and to say welcome to the attendees. As mentioned, I'm a product lead in the therapies for immune and inflammatory diseases, and in the previous life, we worked together in the MHRA as an assessor as well. Thank you.
Thank you very much, Sarina. So it looks like Slido has been already your best friend up to now. It will be your best friend also for the next two sessions. And we can start with the first question. I'm not sure how my colleagues have used Slido, but what I thought of doing is to ask you some questions just to see whether you know the knowledge. The first one is just very general about your background. So that we can tailor a little bit more the session. So we understand that the most of you are working in quality and GMP, but we would like to know whether someone has also knowledge on non-clinical and clinical. Okay, so I mean, this is really was just to check the situation. We don't need exact numbers. Well, it's good to see that there are also people directly involved with clinical and non-clinical, because this topic is in fact multidisciplinary. Okay, so next. So which of the following options best summarize how equivalence of inhalation product is demonstrated in the EU? Is it by totality of evidence, three-steps approach, or by equivalence study? Okay, I think we can stop for this one as well. It's just to have an idea, and also because by the end of this presentation, you should be able to answer 100% the right answer. And to help the group understanding where I'm saying the information, I've got here some very useful tools related to inhalation. And I will, if I remember the right slide, I will raise it. So, so if you're falling asleep, that's the right time to wake up. Second question. How many inhalation guidelines are under revision? Okay, I think we know what the majority of people think. Next one, please. Will hydrofluorocarbon gases used in medicinal products, so also in inhalation products, be banned in the EU? Don't be shy. Okay. Again, a very clear trend. So let's begin. So the title inhalation products is really broad, and today we will cover 4 different topics. So demonstration of equivalence for inhalation product. This was actually one of the topics requested by some of you. Then revision of current guidance documents. We will also talk about the question and answer when replacing hydrofluorocarbons propellants. This is a very recent topic. And we also had an interaction with the WHO on inhaled medicine review. So we will give you a short summary of how the interaction went and what did they find out. So demonstration of equivalent of inhalation products. This is just a table of content, but here already, if you look at step 1, step 2, and step 3, you got an answer for one of the questions. So the main guidance document to use when reviewing equivalence between inhalation products is this one with this very long title, Requirement for Clinical Documentation of Orally Inhaled Products, including requirement for demonstration of therapeutic equivalence between 2 inhaled products for use in the treatment of asthma and chronic obstructive pulmonary disease, back to 2009. Although talks about clinical documentation, there is a section in this guidance, actually section 5.2, which is relevant to everyone working on quality, because these are really the quality requirements that you will need to looking at when looking at equivalence of inhalation products from a quality perspective. So it is a multidisciplinary guidance, and hence also quality experts contribute to this guidance. When looking at demonstration of therapeutic equivalence, we make reference to a reference medicinal product which is marketed in the EU. Normally, this application comes as a bridge application, but other legal aspects are not, legal bases are not possible. It's just that most of the cases, we see them as a bridged hybrid application, so according to Article 10.3, but other legal bases are not excluded. For this Article 10.3 application, a bridge is based on the fact that the bioequivalence cannot be demonstrated through bioavailability studies. And they all have a complete quality data, so a complete module 3, and are supported by appropriate preclinical and clinical data. So in the module 3, what we expect to see is the pharmaceutical development studies. And the pharmaceutical development studies, you will find actually the full description in the quality of orally inhaled and nasal products. It's a different guidance from the one that I mentioned before. And this guidance presents all the tests that needs to be done based on the pharmaceutical form. So if it is a pressurized metered inhaler, a powder inhaler, a product for nebulization. And then it also has the requirements for the specification. Here there are just 2 tables summarizing the tests that need to be presented in the pharmaceutical development. So again, now it is really clear. In Europe, we use what is called a stepwise approach, and we will be going through each steps during this presentation. So step one is pharmaceutical and quality. And so this pharmaceutical and quality, as I said, is summarized in section in the paragraph 5.2. But what is really important to understand is that the pharmaceutical properties are at the base of the evaluation. Even if the applicant might provide a pharmacokinetic or clinical data, we always start with the pharmaceutical quality, and this aspect cannot be waived. This is a flow diagram of how it is possible to reach approvability for a product in Europe when we demonstrate equivalence to a reference product. So as we mentioned before, the first step is the step one, and the answer to the question, are the two products pharmaceutically equivalent? If the answer is yes, by satisfying all the requirements that are provided in guidance, then that's it. The product is approvable, and we do not need to look at clinical data. So we start with the requirement of this step one. The core message is that the use of comparative in vitro data may be considered acceptable to demonstrate therapeutic equivalence if the product satisfies all the criteria mentioned in section 5.2 of the guideline. So all the criteria need to be addressed, even if the application is not based only on in vitro data. And here is a summary of the criteria. So the product contains the same active substance or substances. And when we say the same, we also look at the same salt, ester, hydrate, solvate. The pharmaceutical dosage form needs to be identical, so we can only have equivalence with the same dosage form, so pressurized methadone inhaler, compared with another pressurized methadone inhaler, dry powder inhaler with another dry powder inhaler. Then, in terms of the solid state aspects of the active substance, we need to look at differences in crystalline structure, polymorphic form, and make sure that they do not influence the dissolution characteristics of the product, and also the performance of the product or the particle behavior of the active substance per se. Differences in excipient composition is possible. Generally, though, they make the development more difficult. And these differences need not to influence the performance of the product. So, for example, the delivered dose uniformity, the aerosol particle behavior, for example, the hygroscopic effect, the plume dynamic and geometry. And they also not need to be felt differently by the patient, by the end user. So how these different excipients change the effect in the mouth and the throat, also the cold freon effect needs to be the same. And also, any qualitative or quantitative differences of the excipient should not change the safety profile of the product. So generally speaking, most of applicants tend to use the same qualitative and quantitative composition. Then in terms of performance as well, the inhaled volume through the device should enable sufficient amount of the active into the lung. It needs to be similar. So the inhaled volume needs to be plus or minus 15%. And this year we are talking mainly about dry powder inhalers. Then the handling of the inhalation device for the test and reference product in order to release the required amount of the active substance should be similar. And this is this is slightly different from the requirement, for example, of FDA, where they really want almost a step-by-step equivalence of how the device is used. So if you open, click, inhale, activate, FDA wants exactly the same step, while in Europe we want similar steps. And these are normally looked at also during the human factor studies. Then the inhalation of the device has the same resistance to the airflow. And the target delivered dose should be similar. So here we talk always between plus or minus 15%. Then, interesting, if you look at the guidance, you see these two last points that are not even listed with numbers. And one point says that the aerodynamic particle size distribution should be similar. Again, maximum allowable difference between plus or minus 15%. And this is the point where basically most products do not manage to meet in vitro equivalence, because it's very difficult to have this equivalence in particle size distribution. Another and the last point is that the comparative in vitro data of the flow rate dependence should be obtained with the range of flow rates. So here I've just looked at the pharmacopoeia methods that can be used to determine the aerodynamic particle size distribution. So we have the multi-stage liquid impinger, the Anderson cascade impactor, and the next generation impactor. And these are the ones used to measure the APSD of the products. Anderson cascade impactor and next generation impactor are those that you normally see in most applications. The multi-stage liquid impinger is not any longer so much used. Here is a representation of how the applicant can provide actually the particle size distribution. So it can be in a tabular format like this. They always need to provide the data as well and the data analysis, or they can just be in a tabular format. Obviously, the deposition stages that are more difficult to demonstrate equivalence are those that are with the lower deposition. And that because when compared from test reference product, you might get a variability that is much more than the plus or minus 15%. So this is a representation and is based on confidence interval. And we can see whether they are compliant and non-compliant. And it's not the same data as represented in the graph before, but you normally see these issues of non-compliance in the adapter and in the micro orifice collectors towards the end, and also when the particles diminishes. To demonstrate equivalence, they can also use some graphs, which are, well, make the assessment easier because it's easy to see which one are in within the two bars. And when I was saying about those stages with low deposition, to be like technically fair towards the applicant, we cannot demand that the stages with low deposition are, in fact, equivalent, they meet the criteria. So it is possible to have what is called stage grouping or stage pooling. So group together two non-overlapping groups, two groups that are next to each other. As long as it is justified. In the current guidance, the justification that is given is that it needs to be clinically relevant, but the current thinking is no longer about the clinical relevance, because although the cascade impactors have kind of mimic the deposition in the lungs, they don't really match the deposition in the lung. So we cannot look at the cascade impactor and know where the particles will go in the lung. So what now, during assessment, we are allowed to do is for stages to be grouped when they are one next to the other, and they are very small in deposition. and there are none. And at the end, we'll get at least 4 non-overlapping groups so that they can be evaluated. Okay, so so basically these are the main point for the step 1, the in vitro studies. And so as mentioned before, equivalence can be demonstrated in vitro, but if it can't, then we need to move to the next steps, pharmacokinetic and pharmacodynamic. Independently of whether it is demonstrated in vitro, they still need to look at all these criteria that we saw before. They still need to discuss them all. And then this will give also a good, even if the equivalence is not met in vitro, they give a good indication of how similar the 2 products are. If there are large in vitro differences, even if there is a step 2 and a step 3, maybe that is satisfactory, the applicant really should have considered reformulation of the product. So with this, I finished the step 1 and I give the floor to Sarin.
Hear me well? Can you confirm?
Sorry, Sarin. We are step 2 now. Can you see it?
Yes. Can you hear me well?
It's a bit broken.
Can you hear?
Yeah, I'm not sure. Could we up the volume a bit, maybe?
For if there is something you do from your end?
There is a bit of background noise. I don't know if maybe you use your headphones, it could be clearer, I think.
Okay, I will try that. It works, but let's see.
It's like an echo, really, no?
Yeah.
Yeah, it's a bit of an echo.
Yes, apologies, I will not be with you. Let me see. Now, is it better?
Yeah, much better.
I think that's better, yeah.
Yes, much better. Perfect. I cannot hear you well, so I will try to increase the volume. Sorry for this. Okay. We will go, as Claudia has mentioned, if therapeutic equivalence has not been demonstrated in step 1, we need to move to step 2, to the pharmacokinetic studies. In the next slide, we can see there is a decision tree. We will not go into the details of this decision tree, but we can see that in step 2, we need to demonstrate the equivalence in lung deposition and as well in systemic exposure. So, as mentioned, for the pharmacokinetic study, determination of the total systemic exposure. This is for the purpose of determining the safety. For this, we use the study without charcoal blockade. Charcoal blockade is an approach used to block the gastrointestinal tract, the gastrointestinal absorption for orally inhaled products. And this is helpful to determine the absorption via the lung. That is the intended site of action. However, for safety, we want to understand the effect through all the different absorptions, oral or as well from the lung. That's why we don't use charcoal for the assessment of safety. Determination of pulmonary deposition. This is to assess the efficacy. For this, we consider whether there is negligible contribution from gastrointestinal. A study without charcoal will be sufficient for both efficacy and safety, because if there is negligible absorption following the gastrointestinal tract, it will be the safety as well will be based on the absorption from the lung. However, if there are substances that they have significant oral viability, for example, budesonide
for formoterol, I think salmeterol as well. And in those cases, we will need a study with charcoal blockade. As well, there is the option to have the early partial AUC in a study without charcoal in the case of very quick lung absorption. We mentioned negligible gastrointestinal absorption, but what do we consider as negligible gastrointestinal absorption? Okay, we assume it's low oral viability, but to have a precise determination, there is a definition in the PK Working Party question and answers, which is published in our website. In particular, question 17 is the one focused on the evaluation of orally inhaled medicinal products. And in that question is defined what we understand as negligible gastrointestinal absorption, and it is when it is less than the 5% of the total systemic exposure. We can see in this graph, Claudia, maybe in the, just briefly, I will say a few words on the drawing, that following the invasion, the idea is that it goes to the majority to have some systemic effect, but there is also a proportion that it can be swallowed, and from that can be absorbed into the gut, and there must be as well first pass metabolism. All of these will contribute to the systemic effect, to the adverse effect, and to the safety. Reasons for negligible contribution is because it can be low intestinal absorption or as well extensive metabolism, what is the case of meclometasone, fluticasone, and as well mometasone. Moving, how can it be the study design for these PK studies? Just briefly, because we don't go into much detail for the non-clinical clinical information for this presentation. It will be a single dose crossover study, standard test and reference, sufficient sampling time to allow 80% of the total AUC, and early sampling critical in order to estimate Cmax. As well, we will consider, I will say a few words with the study population. So for the study population, what the current guideline, the multidisciplinary guideline that is currently under consider best, of course, measure PK in the intended patient. However, there is some consensus in the EU that healthy volunteers can be accepted. The reason for this is that, of course, they are easier to recruit and less variable. As well, the healthy volunteers that don't have vasoconstriction may be more discriminative to the effects of the product. And as well, the quality working party question and answer considers the healthy volunteers to be an option where no flight rate dependency for test and reference in vitro, and as well when there is similar flow rate dependency for test and reference. This is determined through the evaluation of the fine particle dose and the aerodynamic particle size distribution. So within for the pharmacokinetic studies, we also need to perform some pharmaceutical in vitro studies to support the in vivo. We will cover. What we need to determine is the choice of representative batches, the choice of strength, those proportionality, and then the flow rate dependency. Claudia will provide information on these in vitro studies.
Okay, and so back with me, and yeah, I always find interesting actually how quality and clinical are really interdependent for inhalation products. Even to give you an example, within EMA, we have what we call matrices that they work on, so for example, respiratory. And for this matrix, usually it's clinical based, but quality is also involved in support of the matrix because of the relevance of the quality aspects. So in fact, these studies that are important for the determination of the type of PK studies, these quality studies are quality working party documents. So they're question and answer that you can find in our website. So in terms of the representative batches, for the reference product, this should be representative of the commercial batches on the market. So several batches needs to be analyzed, between 5 to 6, and as a minimum, and but generally we expect more. And we need to have quite a broad spread of what is available in the European market. And I say different countries because often these products have been first authorized at national level, so maybe either national or DCP or MRP. So there could be some differences. So there are quite old products, and the centralized pathway at the time wasn't an option. So the fine particle dose should be close to the median, and a deviation of plus or minus 15% is considered reasonable. And then the test product should be representative of future batches, and the specification limits are crucial and should be based on the clinical batches. So for example, these are some representation of reference batches, and is a plot, and basically any between these two lines can be accepted. So even if it is not closer to the median, as long as it is plus or minus 15%, is fine and cannot be questioned. So in this case, the black square was chosen as a reference product and is fine for the PK studies. Then another QWP, quality working party question and answer, is the choice of the strength for dose proportionality. So for inhalation products with more than one strength, therapeutic equivalence can be investigated clinically with only one strength, as long as dose proportionality is demonstrated. The all APSD should be compared with an acceptance criteria of plus or minus 50% in a range of relevant flow rate. If not proportional, then a bracketing approach with strength most similar and most different in an in vitro perspective could be acceptable. So if we look at an example, here we have basically the comparison of the APSD between test and reference. We see that the test reference ratio is 0.76 and 0.72. So in this case, we can just choose one strength for the clinical study. Often they choose the strength with either with the lower dose because it's worst case, but then if the APSD determination is more difficult, they might choose the middle or higher strength. In this other case, although if we look at the high and low strength, we see that they sit more or less in the same position as the previous example. If we look at test versus reference, then we can see that it is not possible to waive the use of all to use just one strength for the PK study. And in this case, the three strengths will need to be tested, or if they provide a justification as a minimum, they can have a bracketing of the low and highest. Then for in vitro flow rate dependency requirement with the PK studies in healthy volunteer to investigate the systemic exposure and lung deposition may be acceptable if the test and comparators have shown that FPD, so fine particle dose, and APSD for both test and comparator are either no flow rate dependent or they got similar flow rate dependency. The flow rate interval to be studied should be justified based on the resistance of the device or the trigger threshold for a device that is actuated by like a breath inhalation. And the flow rate observed in healthy volunteers and patient population. Normally, a flow rate intervals between 30 and 90 is generally considered acceptable. So, Sarin?
So here just to, yes, thank you, Claudia. Here is just to summarize the key points of the pharmacokinetic studies to say that although it's recommended to do it in the intended patient population, healthy volunteers can be accepted. The relevance of the selection of the representative batches, selection of the strength, to remember that the studies with and without activated charcoal can be done, whether it's for efficacy, for safety, or whether it has negligible gastrointestinal absorption, and an observation with a study design that early sampling in case of early TMAC. Then I think we move to step 3. For step 3, if the therapeutic equivalents have not been demonstrated in step 2 with the pharmacokinetic studies, we move to the pharmacodynamic clinical studies. We go back to the same decision tree. And as indicated for step 3, we will need to demonstrate equivalence on efficacy and or safety by means of PD and clinical studies. What will we do for these pharmacodynamic clinical studies is that we will cover the aspects only that they have not been confirmed by means of kinetics. So we will do efficacy or safety, or both, depending what it has not been confirmed in the previous step. And it can be done for one or several substances. An appropriate PD model should be sufficiently sensitive and able to distinguish the effect of safety of that particular substance we are studying and we are focusing on. For children, we need to consider that there will be different breathing pattern, different exposure at active site. It might be difficult as well to measure the effect. There might be ethical restraints, whether we could do extrapolation of the studies or whether we expose the children to necessary and unnecessary additional studies. And as well, there are concerns regarding the adequate assay sensitivity in a PD model. So the conclusion, just briefly summarizing 4 bullet points, is that this is always built upon the 2 first. It's focused on whatever has not been confirmed by means of kinetics. The issue of the concern will be the availability to have an appropriate PD model, and as well to consider the difficulties that might be encountered when the study is performed in children.
Okay. So now we move to the revision of current guidance documents. So it was not one, the choice. The hint is in yes. And these are the titles of the documents that are under revision. So the first one is the Guideline on Pharmaceutical Quality of Inhalation and Nasal Products. It's an old guidance from 2006, and that's why there are also a number of question and answer in relation to inhalation products in the IMA website. And the second guidance is the multidisciplinary guidance that we've been looking at up to now, the therapeutic equivalence guidance, which has this super long title. I'm not sure, Sarin, do you know if are we keeping the same title for the revision, because it's really, really long? I think we are going shorter, right?
No, yes, it has been shorter.
So we had 2 concept papers on, well, one was on the quality guideline, and the concept paper outline why there is a need for a revision. So this need for a revision is driven by the need to clarify aspects related to the establishment of therapeutic equivalence based on pharmaceutical data. So the changes to the guideline in the therapeutic equivalence also have an impact on the quality guidance, and that's why also the revision of these two guidance has been proceeding in parallel, so they can be aligned with themselves. And for example, I go to both drafting groups, both to the quality one, and then I support the equivalence one for the quality aspects. And yeah, it's also an opportunity to align the requirements with the oral inhaled product. The main focus of the revision is actually the inhalation part rather than the nasal part, mainly based also on the number of products that we receive and the experience that we have at IMA. So a lot of experience has been gained since 2006 when the guidance was first published. There are new active combination, new chemical entities, and particularly there is also a lot of advantages in inhalation technologies, both like in terms of better drug delivery characteristics. The inhalers have changed, they got those counters, and now they also have some digital functionalities, and they're able to talk to apps and give reminders to patients. But then also the ways that they are analyzed, they are becoming more automated and sophisticated. So all these aspects will be taken into consideration in the revision of the guidance. We received quite a few comments, and in terms of the comments, there were a lot of device-related topics. So data requirement for spacer and holding chambers, how will this be reviewed in the quality part? As we highlighted, consistency with the equivalence guidance, but also with the drug device combination guidance and the MDR. And particularly with the MDR, we do receive a lot of queries that some of them actually do fall outside the IMA remit, so about the medical device classification. But it's important that the revised guidance will take these aspects into account. And the other topic is on actually e-connectivity, and it's quite interesting because it sits in the module 3 under device most of the time, or in the regional part of the module 3. So what requirements are needed if an inhaler has an e-component, so a sensor that sends data to an app. So that's quite a new and interesting and also challenging topic. And then another point that we received is the need to align patient handling studies with current practice, so human factors. So the guidance has been under review for a long time, but it was suspended because of business continuity plan. So first IMA moved from London to Amsterdam, then we had COVID. So now we are back again working on what we say that is not non-core business activity. And we've been busy looking at all these aspects again. UK is no longer a member, so it was actually someone from when I was an assessor at the MHRA that has been a long-standing member of the drafting group, but it can no longer participate. Then the revision of the guidance has been agreed by the Quality Working Party for the 2023 work plan, which was adopted by CHMP in December 2022. The target date for the finalization of this of the revision guidance is at the end of 2023. Maybe we can make it, let's see. It's quite ambitious because there are several topics that are under revision. But we'll strive to do it at the same time as the clinical equivalence guidance. Now, Sarine.
Yes, thank you. The revision of the equivalence guideline, the first version is dated 2004, quite a long time ago. The first revision is the current version that is dated 2009 and is the known multidisciplinary OIP guideline. This is the one that is currently under revision. It was affected as well by, of course, the Brexit and the pandemic. Revision 2 is the concept paper on the revision of the guideline. And if we can see here, it explains the topics that they needed to be revised. And it was to provide, because it's currently used, the step wise explain, but to provide more details. This approach is opposed to the FDA, that is on the totality of evidence. They don't go step by step. They just evaluate the whole data package. The revision of information on job for batches, the strength and study population, the PK studies will be reviewed. It will also cover for advances in inhaler technology, and as well data requirements for the pediatric population will be discussed and reconsidered. And for this, we are already in contact with the PDCO, the pediatric committee. So the timeline, the revision, it was agreed in the 3-year work plan of the Rheumatology and Immunology Working Party that was adopted last December, and the target date is as well that the draft revised guideline to go for public consultation by the end of this year.
Okay, and
then I think this is the
sorry.
this is the third topic.
Yeah.
Can you hear me?
Yeah.
Yes. So this is the third topic for today, is the question and answers when replacing HFC propellants. It's a document that it has been recently published and has gone through 2 months consultation, and it's available in our website. If in the previous slide, you can see what we will cover in bullet points, just briefly the introduction and general principle, the requirements, and as well the specific considerations related to the product information. So just briefly, as background, as you know, pressurized metered-dose inhalers, they use HFC as propellants. However, HFC have high global warming potential. There is a legal definition for this, but basically is the climatic warming potential of a greenhouse gas relative to that of a carbon dioxide. Fortunately, there are different alternatives. It can be considered that the active substance can be used in a different pharmaceutical form. That might not be the ideal approach, mainly for the patient that they need to adjust. And the other option as well will be to have the same pharmaceutical form, the same pressurized metered-dose inhalers, but replacing the propellant with a low global warming potential one. What is the international context? The Montreal Protocol, it was international treaty. It was agreed in 1987, and it was on the ozone depleting, and it was also covered in the HFCs. So it was agreed that since 1989, the ozone hole recovery expected in second half of century, and that since 2019, it was also considered the phasing down of the HFCs. The protocol contribution to the other international treaty, the Paris Agreement dated 2015, it was to have a maximum increase in the global average temperature of 2 degrees Celsius, ideally only 1.5 by the end of the century. It covers global ODS, and for what it mostly concerns to us, it will be for the global. This is to aim avoiding around 0.4 degrees Celsius increase by the end of the century compared to the historic triggering regulation. In April last year,
we cannot hear you, Serene. Can you hear us? I think there are some issues with the.
Yes, I can hear you.
Okay, I think there are some issues
Internet?
with your Wi-Fi, I think.
I'm okay. It seems to be fine. I don't know if you can hear me now. I removed, there is no video. Can you hear me?
Now we can hear you, yeah.
Yes.
Okay.
And I think it
Okay.
would be good to keep the
So
video
without
off.
the camera?
Yeah, no, just keep
Yes,
it off,
yes,
I
yes.
think.
It will be like that. I will do. So the proposal it has been to update the regulation to regulate the use of the higher global warming potential HFCs in the PMDI sector, and to also include the supply of HFCs to the PMDI production under an existing HFC quota system. So it's not to ban from one day to the other, but to reduce the availability in the EU over time. Another hint as well, like Claudia gave earlier. So we can go now to the data requirements that they are covered in these questions and answer. Claudia, the quality?
Yeah. Before looking at the quality data, I also just wanted to say that for this topic, it's actually had a really, really big impact for, has a really big impact for the manufacturer of inhalation products. Because a change in propellant is not an easy change. Some people think, oh, well, you're just changing an excipient and it's going to be very straightforward. But for inhalation product, it changed the performance significantly. So, and although it would fall just under a quality variation, a quality change, is actually normally supported by clinical and non-clinical data. Or PK/PD data, and hence is a very expensive change. So we had a lot of scientific advices, a lot of interest. It's been quite a critical topic coming to us. And the applicants have actually received very well the very prompt publication of this question and answer. And in fact, among the big regulatory agencies, we are the first one. And it's been developed by also sharing information with other regulatory agencies like FDA and Health Canada. So there is a lot of work that has gone into it, and there's quite big significance for applicant. So the first part,
Indeed.
Yeah, if you want,
Claudia,
chip in. Yeah.
No, yes, sorry to interrupt you. No, yes, thank you. Because I realized I did not cover, and it will be done briefly, the previous slide, is with regards to the triggering regulation. Just briefly, that the European Parliament and the Council, they have adopted their position. This was done during in March this year. And this is the starting point for the final compromise legal text that is expected to be adopted by the second half of this year, and it may already apply from January next year. Just this piece of information that I thought it was relevant. Thank you, Claudia.
Okay. So for what concerns quality data requirements, in view of the potential impact of the propellant on the functionality and performance of the product, we need to consider pharmaceutical development studies. There are specific considerations for patient population, so expelling pressure, taste, feeling in the mouth, and flammability. So flammability is a new aspect that comes with new propellants that are very flammable, they're difficult to manufacture, they're difficult to store. So that's another challenge that actually manufacturers have. Then there is a reevaluation of the finished product specification at release and then the shelf life. And also a discussion and justification of device-related changes. So for example, components of the valve and canister, not only for quality aspects such as extractable and leachable, but really to adapt the device to the new propellant. Manufacturing method validation, since they're not standard manufacturing method use, and stability data also need to be provided. Then Sarine, moving to non-clinical data. Yeah.
Thank you. Yes, for non-clinical data requirements, primary and secondary pharmacology studies are not expected because there is no pharmacological activity expected for an excipient. For a novel excipient, is considered that it has not been previously used in any approved medicinal product with the same route of administration. So for those, the non-clinical requirements will be limited to toxicology and pharmacokinetics. Safety pharmacology data might be obtained in toxicology studies obviating for the standalone safety pharmacology studies. This could be accepted, but always needs to be justified in the dossier. Guideline on the excipients in the dossier for application for marketing authorization applies as for any new substance. And to excipients in a pediatric population, the ICH S11 may also apply. So for only when there is the propellant is a novel excipient is when the full data, the safety, the data requirements on local tolerance and data requirements on clinical safety will be needed. If it is already an established propellant and established excipients, these data requirements will be significantly reduced. So for novel, the data should be generated by two studies, ciliary function and airway sensitivity reactions. These studies are new. As Claudia has mentioned, we are in contact with the company, scientific advices, and this is what the main two studies that they have been presented and they have been considered to be suitable. However, they are new to the regulatory settings and there are uncertainties regarding the choice of primary endpoints and the non-inferiority margins. So the really strong advice from the agency is for the companies to come and request scientific advice, and then we'll advise them and guide them on their development. Just briefly to say on the clinical safety that at least should be 1 3-month safety study, main objective to collect the adverse events. For the test, the vehicle version of the final formulation is accepted, and even as it is considered it will be for 3 months, it could be also acceptable to use a finished product formulation indicated for daily maintenance treatment. We will also need, compared to product, it is recommended, as it mentioned in the following slide, that we need to choose product strength where the number of actuations needed is in the higher range. And to mention again that this 3-month safety study provided in the question and answer is just descriptive, and there are no defined endpoints and non-inferiority margin given at the moment. We are all learning through the process. So in addition, when there is a replacement of the HFC propellant, we have quality data to be provided, non-clinical safety tolerance when there is a novel excipient, but of course we need to demonstrate and to keep that is therapeutic equivalence. This will be approach, and it should be done for each product, each strength of the product that needs to be varied. Therapeutic equivalence will be confirmed as outlined in the guideline, the multidisciplinary guideline that we have mentioned earlier. It should be provided with both with and without a space or holding chamber. For hybrid or generic, the comparator should be the reference product, the reference medicinal product that it was used at the time of approval, irrespective of the propellant used, even if it has not replaced the HFC. The data requirements are fully in line with the OIP guideline that is currently under revision. So in the next slide, we can see the 3-step, the ladder, and as well, instead of the decision tree in a flow chart. So in the next 2 slides is summarized.
Sorry, we cannot hear you. Do you? Basically, the 3 steps approach are the same that we described before. Sarin, can you hear us? If not, I can.
Yes, can you hear me now?
Yeah, now we can. Yeah. But I don't think we need to go in. Do you want to go into the details? For the quality, the pharmaceutical aspects is basically the same that we covered before. And again, data needs to be provided. Sarine, do you want to cover step 2? Maybe she cannot hear us, or maybe we cannot hear her. But anyway, then we have the same steps that we saw before. And in fact, the question and answer is not is detailed on what is new about the change of the excipient, but otherwise it makes reference to the inhalation guidance, the equivalence guidance, because whatever is going to be changed there, it will also be applicable for change in propellants. So the guidance refer to each other. And Sarine?
Have explained already. Can you hear me now?
Kind
We
of.
know requirements, data requirements for children and adolescents.
Yes, go for that. Go.
Yeah. You hear me well? If not, you can.
And not so well.
Maybe you can give it another try, Sarine.
Extrapolate. Yes, I will try. So for children and adolescents, the extrapolation from studies supporting safety of a novel propellant in adults to children and adolescents is acceptable. If the therapeutic equivalence between the test and the reference is confirmed at step 1, the same age limit as for the reference can be applied. Extrapolation to the conclusion of the therapeutic equivalence demonstrated by PK studies to adolescents as long as the reference is approved for this same age group can also be accepted. And it may be acceptable as well to keep the same age limit currently approved for the product, subject to the change of propellant.
Okay, and
for you, Claudia?
Yeah, we'll cover now some aspect of the product information. But I also wanted to say on the previous slide for the requirement for children, the drafting group will really try to facilitate maintaining the same indication as the reference product and reducing to the minimum the number of studies in children because they're difficult to be recruited, conducted, there are ethical reasons and so on. So now looking at the product information, so the name of the product, changes in the qualitative composition or quantitative composition of the excipient do not require any change in the name product. So we do not expect to see a name that changes because the excipient has changed. Then inclusion of statements such as FC-free on the label is not supported because we see that there is no reason to state what is not present in the finished product. In fact, it can be seen as promotional, and that should not be allowed. We talked before about flammability warning, and if necessary, that can be included. And there are some wordings in the EC guidance on flammability, and so something similar can be taken into consideration, and it will be reflected in the SMPC in section 4.4, and then also in section 2 of the packaging leaflet, and the name of the product contains whatever the excipient in question. Special storage condition might be required. So, for example, to keep under a certain temperature, not to pierce, and so on. And this should be stated in 6.4 and section 9. And so that concludes the part on the change in propellant. And now we have a few last slides on the interaction the EMA had with the WHO. The WHO was preparing a desk review mapping of the global manufacturing regulatory landscape of inhaled products for the essential medicine list for asthma and COPD. So 2 consultants from the WHO came to IMA for an interaction, and came virtually, they got in touch. And so the background information on this consultation is that asthma and COPD are considered 2 of the main chronic respiratory diseases included in the WHO Global Action Plan for the Prevention and Control of Non-Communicable Diseases. The action plan spans till 2030. And the effective treatment of asthma and COPD relies on the availability of medicine, both for emergency and long-term management. However, access to safe, effective, quality assured, and affordable inhalers is a problem in a lot of countries. So the WHO Global Action Plan for the Prevention and Control of NCDs includes a target of 80% availability of essential medicine required to treat major NCDs in both public and private facilities. The objective of this desk review was to map the global manufacturing and regulatory landscape for inhaled medicines on medicines that are listed on the essential medicine list. So to identify products that are registered, they came to some stringent regulatory authorities. That is meant like USA, Canada, Australia, UK, European countries. In this case, I was TGA, Swissmedic, and EMA that were selected. And the aim was to identify where the sources are registered and also in middle-income countries, and identify sources being procured and used by UN agency doing procurement of health products, and see if there were any obstacles for the availability of these medicines. So the key finding were that all selected procurement agencies do procure at least 1 bronchodilator and 1 inhaled corticosteroid. However, it does not ensure adequate availability rate at health facilities in countries where these procurement agencies operate. The WHO essential medicine list presents some inconsistencies. So in the recommended of strength and specification, and so there are challenges for national procurement agency to procure relevant inhaled medicines, and prescriber not knowing what dose is delivered to the patient, so they do not know what replacement they can use. So they highlighted the lack of international harmonization with regards to regulatory requirement, where we see that in Europe, for example, we have the 3-step approach for totality of evidence used by FDA. Expression of strength, delivered dose versus emitted dose, is also a big topic. We also see it at European level because of the old products that are approved on the market and that we keep consistency, but then the new products are in terms of delivered dose, and also differences in pharmacopoeias. So the increased complexity for the management of the product dossier, labeling, and packaging has an impact also on the supplying of to different territories. So this is a topic that does come quite frequently about discrepancies between territories. And with this, our presentation is finished. Let's see if those 3 questions, at least those 3 questions, are now clearer. Clement, if you can help me with the Slido. I'd be very upset if he hasn't improved. So which of the following options best summarize how equivalence of inhalation products is demonstrated in the EU? Is it totality of evidence, 3-step approach, or bioequivalence study? Okay. I think we can stop it here. Yeah, so it's getting even better. Maybe now we keep on going. So it looks like that less than 10% of the audience was asleep during the presentation. So that's a great result, particularly after lunch. Next question, please. How many inhalation guidelines are under revision? All right, perfect. So same result, the same people asleep. Very good. I think there were nine in the room. I was looking if anyone was nodding off. Last question. And this is a bit of a tricky question.
Variation, by the way. Given part of there, that's one aging. I know it changes the whole S part. I thought it was.
Could you please mute your mic if
I
you're
don't.
on?
Yeah.
Alta? Okay, so will hydrofluorocarbon gases used in medicinal product be banned in the EU? And yes is actually the closer answer to the truth. None of two are true, really, because they're phased down. So there still will be a tiny little quota that is allowed for medicinal products, but then because manufacturers are they don't have any real profit in manufacturing for the other industry, they're going to be disappearing also for inhalation products. So well done for following the presentation. And yeah, have we got time for questions?
Yes, I'm looking both in the room and online. Thank you, Claudia. Great presentation. I think that was quite effective, looking at those last three results. I see there's a question from Snezana Nikolic. She asked us to give us more details about biowaiver expectations for additional strengths. Is grouping of stages possible in biowaivers as well?
Grouping of stages possible. Well, okay, so grouping of stages is under current active discussion. When is it possible? How is it possible? What criteria? So has it been possible in a current marketing authorization application? The answer is yes. So if there is a test and reference product and we look at the stage comparison of the APSD, grouping of stages is possible. Under which conditions? So how can they be justified? That is under discussion. So I would say watch this space, and so you will get a definite answer rather than just my personal opinion. I've got a feeling, but I'm not the representative of the experts. So yeah, I hope this answered the question.
All right, thank you. I see our colleague from Bosnia. If you could please state your name and your question.
Hello, I'm Realizovic Agency for Medicine and Medical Devices of Bosnia and Herzegovina. I'm interested in the position of EMA about in silico methods of determining bioequivalence, especially in inhaler products.
So there are quite a bit of discussion. Rather than in silico, at the moment it's been more dissolution, whether some dissolution method with some specific validation, they can be considered as a surrogate for either PK or PD. In silico, we have had, I think, one ITF meeting. But whenever you write a guidance document, you need quite a weight of evidence to include reference to something that is novel. And in fact, the idea is that it would be good if applicants came over with the traditional approach, supported and matched up also by a novel method. So dissolution or in silico, to demonstrate then that actually is robust enough to waive some aspect of the requirement of the guidance.
Thank you, because in these guidelines, in silico methods are not mentioned at all. But developers of those softwares are bragging that they are accepted in FDA and EMA.
Well, so from my experience, I haven't seen any application that doesn't follow the three-step approach. We have interaction, as I mentioned, but in terms of guidance document kind of endorsing an in silico method for demonstration of equivalence, no. We have actually qualification of software, but that's more for clinical endpoint and is a different kind of so is a different part of not even an application, it's a different procedure altogether. Yeah.
I think, Claudia, if there is time, we received some questions in writing before the session. Maybe we can cover them briefly.
We still have a few minutes, so if you want to, and there are no
Yes.
at the moment, yes.
Should I start with the first one, or you go, Claudia, if you have them there?
Well, the first one, basically, it was mainly the main topic of the presentation, right? Bioequivalence it was for dry powder inhalers, but what we saw, it was applicable for PMDIs and dry powder inhalers. So the requirement for in vitro equivalence are the same. If it is a dry powder inhaler or if it is a PMDI, we go back to the multidisciplinary guidance, section 5.2, and there are all the requirements that need to be demonstrated. It's easier for PMDIs than for dry powder inhaler to demonstrate equivalence because of the aerodynamic particle behavior for a powder or for a pressurized method-dose inhaler, but the requirements are the same. Then, so that was question 1. Question 2.
Maybe to read the question for everyone to understand.
To the best of your knowledge, were there any cases where equivalence to a reference medicinal product was accepted based on a comparative in vitro data only, even if some of the criteria defined in the guideline were not met, but the justification from the manufacturer was provided? Well, I hope that after this training, it's clear that all the points needs to be demonstrated. Otherwise, you need to move to step 2. So we think there are no cases. Obviously, we cannot speak for national competent authorities, maybe, but at EMA level, if not all the steps are demonstrated, all the points are met, then you need to move from step 1 to step 2. Shall I go for question 3? How does EMA evaluate the similarity of inhalation devices between the test and the reference product? So hopefully I covered that also during the presentation. So they have to be similar in terms of functionality, in the sense that if it is a pressurized method-dose inhaler, you're expected to press and emit the dose like the reference product. For dry powder inhalers, these are a little bit more complex in terms of the steps and so on. But as I mentioned, in Europe, we do not want a step-by-step exact match. So the dry powder inhaler device could look different. So if we are trying to develop a hybrid of like, for example, the discus inhaler, which is round, you open it and then you inhale, you could have something different. You could have something that looks not round. And in Europe is accepted. FDA might question it because they really want a patient to have the same kind of experience. Then all the requirements of under 5.2, so airflow resistance, and yeah, they need to be met in terms of the device, and then there are the human factor studies that need to be provided. Sorry, we wrote a very long answer.
The other questions are with regards to the guideline that is currently
but-
under review as per. So I think we have all covered all the questions.
Then there is the one about the excipients, and yes, they can be like, so they ask, can there be a small change in excipient? So, yeah. And so, for example, addition of magnesium stearate, for some dry powder inhaler might contain magnesium stearate to change the flow properties. Is it accepted? Yes. But it needs to meet all the criteria, so then there cannot be a change for the APSD. There cannot be a change for the emitted dose. There cannot be a change also in the patient field, but in this case, most likely it's not relevant. So yeah, so basically in line with guidance. Okay, this is then again.
I think that questions have been covered. Yes.
Yeah. Okay. And then the last question. I think this was about clarification of section 5.2 of the equivalence guidance. And we hope we cover it during the presentation, and hopefully the concept, after the revision of the guidance, is going to be even clearer for the reader.
All right, thank you. So I see we're 2 minutes into our coffee break now. If there's one last question in the room or either online, then we can still take it. Otherwise,
you can come to me.
Come to us. Yeah, come to Claudia, not to me. Come to Claudia in the coffee break. So no, otherwise, thanks a lot, Claudia and Sarine, for providing this very comprehensive presentation.
Thank you.